Microbial Matters, a podcast introducing the latest microbiome research. Hi, I'm Jerry Zhang, a chemical biologist working on human microbiome. In this podcast, I'm going to share with you newest advances in microbiome research, especially those related to human health. In recent years, more and more studies have shown that the imbalance or abnormality of intestinal microbiota is related to the occurrence and development of some metabolic diseases such as obesity, diabetes, hypertension, hyperlipidemia, etc. This reminds me the classic study on the relationship between intestinal microbiota and obesity. Jeff Gordon's lab at Washington University in St. Louis wanted to study why some identical twins can be very different in their body weight. For example, one obese and the other not, when their genomes are almost exactly the same. So they collected fecal samples from these twins who were different in their body weight and transferred them to genetically identical germ-free mice. They were surprised to find that mice that received gut bacteria from the heavier twin gained more weight than mice that received gut flora from the lean twin. This study confirmed that gut microbiota as an environmental factor has an important impact on body weight and metabolism. When it comes to metabolic diseases, I have to mention the type 2 diabetes. With the modernization of lifestyles, the incidence of type 2 diabetes is rising rapidly globally. And according to CDC, 8.6% of US adults have diagnosed type 2 diabetes, which represent 21 million adults. In recent years, studies have also found that the gut microbiota has a significant impact on type 2 diabetes. Gut microbes can affect the occurrence and progression of type 2 diabetes through a variety of ways. For example, it can decompose carbohydrates in food to produce some short-chain fatty acids, which can stimulate the secretion of intestinal hormones such as glucagon-like peptide 1 or GLP-1, etc., thereby enhancing insulin sensitivity and secretion and reducing blood sugar levels. Second, gut bacteria can affect the integrity of the intestinal mucosal barrier, resulting in increased intestinal permeability, allowing some bacteria or bacterial products to enter the blood circulation, causing a systemic low-grade inflammatory response. This inflammatory response can damage islet cells, reduce insulin secretion, and interfere with insulin signaling, leading to insulin resistance. In addition, intestinal flora can also affect lipid metabolism and produce some metabolites such as bile acids, lipid polysaccharides or LPS, triglycerides, etc. These metabolites can affect insulin sensitivity and secretion through different mechanisms. To sum up, the gut microbiota has an important influence on type 2 diabetes. Therefore, modulating the gut microbiota may be an effective strategy for the prevention and treatment of type 2 diabetes. Patients with type 2 diabetes need lifelong use of drugs to control blood sugar. The most commonly used drugs include metformin, insulin and its analogs, alpha-glucosidase inhibitors, etc. In addition, there are some new hypoglycemic drugs such as DPP-4 inhibitors, GLP-1 receptor agonists, etc. The magic drug metformin has been demonstrated by many studies that it can significantly affect the human gut microbiota, especially the enrichment of akkermansia, which is associated with the reduction of systemic inflammatory response. But the gut microbiota can also mess with hypoglycemic drugs. Today, I will introduce 3 recent studies in this regard. As mentioned earlier, alpha-glucosidase inhibitors such as acarbose are commonly used hypoglycemic drugs. Its function is to competitively inhibit glucoside hydrolases in the intestinal tract, thereby reducing the rate at which polysaccharides and sucrose are decomposed into glucose, and slowing down the absorption of sugar accordingly. So it has the effect of lowering blood sugar after meals. However, doctors have found that the effect of this drug varies greatly from person to person, and some patients don't even respond to the drug at all, or develop serious drug resistance after long-term use. Labs from the Chinese Academy of Sciences and others recently published an article in Nature Metabolism explaining the role of intestinal flora in it. They used the microbial enrichment method to isolate the intestinal strain Klebsiella greenmontii TD1 that can metabolize acarbose. This bacterium has a previously unreported glycosidase that can metabolize acarbose, which can degrade it into small molecular products containing tricyclic and bicyclic structures, thereby making it lose its drug function. This glycosidase and its homologs are widely found in intestinal microorganisms, especially Klebsiella species. Further metagenomic analysis found that the abundance of this bacterium was higher in the intestines of patients with weak acarbose response, and its abundance tended to increase with the long-term usage of this medication. Therefore, for patients with acarbose resistance, future research can consider strategies to reduce the occurrence of drug resistance by regulating the bacterial ecosystems in the gut or targeting their metabolic enzymes. I also mentioned DPP-4 inhibitors as antidiabetic drug. DPP-4 is a serine hydrolase in the human body. It can degrade GLP-1, and GLP-1 can stimulate insulin secretion and reduce glucagon. Therefore, inhibiting DPP-4 can allow GLP-1 to stay in the body longer and maintain blood sugar stability. So what effect does this oral drug have on the gut microbiome? There's a very common bacterium in the intestinal flora called Bacteroides thetaiotaomicron, or B-theta for short. It has many serine hydrolases in its genome, but their functions are still unclear. Recently, Matthew Boggio's lab from Stanford University and others published an article in Nature Chemical Biology. They used a fluorophosphate-based chemical probe that selectively reacted with serine in the active site of the enzyme, followed by affinity enrichment and mass spectrometry identification, thus discovering 27 enzymes that reacted with fluorophosphate. Through bioinformatics comparison, 6 of them are similar to human DPP-4 protein. A series of subsequent biochemical experiments proved that 2 of these proteins are serine hydrolases, which can cut off 2 or 3 residues at the end terminus of the peptide, and the enzymatic activity of one of them can be inhibited by various hypoglycemic drugs targeting DPP-4. What effect do these 2 proteins have on the bacteria? The authors found that these 2 proteins are in the periplasm of B-theta, and they can help the bacteria resist some external stress on the outer membrane, such as bile acids. If they were knocked out, B-theta would also be less abundant in the population. In conclusion, this article combines the methods of bioinformatics and chemical proteomics to identify a protein similar to human DPP-4 in gut bacteria, and studied its role in maintaining bacterial physiological functions. However, the effect of DPP-4 inhibitors on the overall composition and function of gut microbiota needs further exploration and research. See, just in August 2023, the teams from the School of Basic Medical Sciences and College of Chemistry and Molecular Engineering of Peking University, as well as other labs from China and from US NIH, collaborated to publish an article in Science titled Microbial-host isozyme analysis review microbial DPP4 as a potential anti-diabetic target. For the first time, they proposed the new concept of microbial host isozymes, that is, enzymes in gut bacteria with the same function as the host's enzymes. They found that microbial host isozymes are widely present in the gut and can mimic the functions of host enzymes, and they participate in the development of diseases. The research team first evaluated the activity of isozymes in the fecal flora of healthy volunteers and found that 71 isozymes were present in the intestinal flora. Among them, they found that the activity of the bacterial isozyme of the host's DPP4 was the most stable and common among different individuals. However, the DPP4 of the gut microbiome is mainly in the intestinal lumen. So, can the bacterial DPP4 come into contact with the GLP-1 of the host, thereby exerting the similar function of the host DPP4? The researchers found that under the condition of long-term high-fat diet or chemical substance-induced intestinal barrier damage, the bacterial DPP4 can enter the intestinal tissue, significantly reduce the level of active GLP-1, and induce abnormal glucose tolerance. Intestinal barrier damage is a common pathological feature of a variety of metabolic diseases, including type 2 diabetes. Therefore, bacterial-derived DPP4 can enter the host under the condition of intestinal barrier damage and thereby induce abnormal glucose tolerance across the kingdom. Subsequently, through the isolation and cultivation of gut strains and the detection of DPP4 activity, the researchers found that many species of Bacteroides have bacterial DPP4 activity, among which the DPP4 activity of B theta is the highest. It's you again. And it's even more active than human DPP4 to efficiently degrade GLP-1. This result corroborates with the article in Nature Chemical Biology. Sitagliptin is a DPP4 inhibitor that is an antidiabetic drug. It's the second best-selling antidiabetic small molecule drug in the world in 2021. However, clinical studies have found that sitagliptin cannot effectively lower blood sugar in about 40% of patients, and the reasons behind it is not clear. The researchers found that sitagliptin and other therapeutic drugs developed against human DPP4 have relatively weak inhibitory effects on DPP4 of intestinal bacteria. They further recruited 57 patients with newly diagnosed type 2 diabetes and conducted a 3-month intervention study with sitagliptin. They found that patients with low response to sitagliptin had more B theta in their gut. And the animal experiments also found that DPP4 by B theta affects the clinical effects of sitagliptin, which provides a mechanistic explanation for the individual differences in the clinical response of sitagliptin. According to the results above, the bacterial DPP4 is a new therapeutic target for type 2 diabetes. Therefore, the researchers established a high-throughput drug screening system. Through the high-throughput screening of about 107,000 small molecules, they discovered an active ingredient in the traditional Chinese medicine named dorasolin has the strongest inhibitory activity on bacterial DPP4, but has no inhibitory effect on host DPP4. After further structural modification, the researchers determined that DAL-D4, a derivative of dorasolin, is a potent and highly selective bacterial DPP4 inhibitor. In animal experiments, DAL-D4 can improve glucose tolerance by inhibiting gut bacterial DPP4. At the same time, DAL-D4 has no obvious effect on the growth and composition of intestinal commensal bacteria. To conclude, The specific inhibition of bacterial DPP4 can effectively improve the glucose metabolism disorder in mice without disturbing the overall function of gut microbiota. Compared with the article in Nature Chemical Biology, this work mainly explores the effect of bacterial DPP4 on the host, while the other article mainly evaluates the effect of beef theta's DPP4 on beef theta. These two papers complement each other, as the important role of bacteria-derived DPP4 is studied from the perspectives of both symbiotic bacteria and hosts. I like the last article more because it not only reviews the molecular mechanisms, but also discovers and invents a potential new type of antidiabetic drug, which comes from plant extracts. Moreover, it uses modern medicinal chemistry to transform and upgrade the compound to achieve better results. I hope it enters clinical research soon and benefit more patients with diabetes. Cool, that's it for today. Thanks for listening. You can listen to more episodes in your favorite podcast platform like Spotify, Google Podcasts, and Apple Podcasts. You can also leave your comments, it'll really help my podcast. You can also support my podcast, Microbial Matters, on Anchor. Thanks again.